Welcome to Fun Facts Online. Imagine two snakebite victims. Both rushed to a hospital in the dead of night. Both were bitten by the same species, the formidable Mojave rattlesnake. Logic dictates they should receive the same treatment. Yet, one patient develops rapid, terrifying paralysis, their breathing muscles failing, while the other suffers from massive swelling, excruciating tissue decay, and uncontrolled bleeding. The same snake, two drastically different results. How is this possible? The answer lies in one of modern biology's most fascinating fields, venom geography. The venom of a snake is not a static chemical weapon. It is a dynamic, evolving cocktail, and its composition is powerfully shaped by the snake's address. From the arid deserts of the American Southwest to the remote islands of Australia, a snake's location dictates its diet, its predators, and its environment. These local pressures sculpt its venom at a molecular level, creating a life-or-death geographic mosaic that complicates snakebite treatment and challenges the very foundation of how we create antivenom. Today, we're taking a journey into this hidden world of rapid evolution, where a snake's toxin is a liquid reflection of the land it calls home. Before we explore its geographical variations, it's essential to understand what snake venom truly is. Far from a simple poison, venom is one of the most complex secretions in the natural world. It is a highly sophisticated arsenal of biologically active molecules, primarily composed of proteins and smaller chains of amino acids called peptides. Some venoms can contain over 100 different proteins, each with a specific role. The primary purpose of this potent brew is not for defense. It evolved principally as a tool for predation, to rapidly immobilize, kill, and even begin digesting prey. Defense is a secondary, albeit effective, application. These toxins can be broadly categorized by their devastating effects. First, you have neurotoxins. These target the nervous system, either by preventing nerves from communicating with muscles or by blocking the receptors on the muscle cells themselves. The end result is a swift paralysis that can lead to respiratory failure as the diaphragm ceases to function. The venom of mambas, cobras, and kraits are infamous for their potent neurotoxicity. Next are the hemotoxins, which wreak havoc on the blood and cardiovascular system. Some are procoagulants, causing blood to clot so rampantly throughout the body that clotting factors are depleted, leading to uncontrollable bleeding elsewhere. Others are anticoagulants, preventing blood from clotting at all. Many viper venoms are rich in hemotoxins, causing internal bleeding and circulatory collapse. Then we have cytotoxins. These toxins cause cell death or necrosis. They are responsible for the horrific local effects seen in many snakebites: extreme pain, swelling, blistering, and the progressive decay of muscle, skin, and bone around the bite site. Finally, a specialized type of cytotoxin, myotoxins, specifically target muscle tissue. They cause muscle fibers to break down and release their contents into the bloodstream, which can lead to kidney failure. A single snake's venom is rarely just one type of toxin. It's the precise mixture and abundance of these different protein families that determines its overall effect, creating a unique toxicological signature for each species and, as we're about to see, for different populations within that species. This brings us to the groundbreaking concept of venom geography. Think of it like regional dialects of a language. The core vocabulary is the same, but slang and pronunciation can vary dramatically from one place to another. For snakes, these dialects are written in a language of proteins, with life or death consequences. This phenomenon is driven by the fundamental principle of evolution, natural selection. A snake's venom must be optimized for its specific circumstances. The most significant driver of this variation is, overwhelmingly, diet. This is often called the prey-driven hypothesis. A snake develops a toxin that is particularly effective against a local mouse species. Over time, some mice may develop partial resistance. This, in turn, puts selective pressure back on the snake to evolve a modified toxin to overcome that resistance. It's a relentless co-evolutionary arms race. This dynamic explains why venom can change so dramatically over relatively short distances. A population of snakes on a mainland, feeding on frogs, will have venom optimized for amphibians. If a group of these snakes colonizes a nearby island where the main food source is nesting seabirds, evolutionary pressure will favor snakes whose venom is more effective against avian physiology. While predation is the primary driver, defense also plays a role. The evolution of spitting in some African and Asian cobras is a prime example. This is a purely defensive adaptation aimed at deterring large threats, including our own primate ancestors. This defensive pressure selected for venom with higher concentrations of pain-inducing toxins, making the venom a more effective deterrent when sprayed into sensitive eyes. To truly appreciate the impact of venom geography, we must look at the snakes themselves. No species better illustrates this than the Mojave rattlesnake of the American Southwest and Mexico. For decades, scientists were puzzled by the wildly different symptoms of its bite. Today, we know this is because the species possesses two primary, dramatically different venom types. Venom type A is the infamous neurotoxic venom. Its lethality is driven by a potent neurotoxin complex called Mojave toxin, which leads to systemic paralysis and respiratory failure. A bite from a type A Mojave is characterized by delayed symptoms and minimal local tissue damage, making it deceptively dangerous. Venom type B is primarily hemorrhagic and proteolytic, meaning it destroys tissues and disrupts blood clotting. It lacks Mojave toxin, but is rich in enzymes that degrade blood vessel walls. A bite from a type B snake causes immediate, severe pain, massive swelling, and extensive tissue necrosis. The geographic distribution of these venoms is a fascinating mosaic. Type A is predominant in much of the Mojave's range, while Type B is concentrated in specific regions, particularly south-central Arizona. Even more intriguingly, in the zones where these populations meet, snakes with venom type A plus B exist. These hybrids possess both the neurotoxin and the hemorrhagic enzymes, producing a doubly dangerous cocktail. Let's travel to India, to the Russell's viper, a snake responsible for more snakebite deaths there than any other species. This is due not only to its potent venom, but also to the extreme variation across the subcontinent. A 2021 study revealed that the venom of Russell's vipers from 5 distinct regions of India were remarkably different. Snakes from northern and western India have venom that can be twice as toxic as that from southern populations. This means a bite in one part of India might cause predominantly neurotoxic symptoms, while a bite in another region leads to kidney failure, and in a third, severe blood clotting disorders. Now, let's go to Australia, where the tiger snake provides a perfect natural experiment. On Chapel Island, these snakes are much larger and prey almost exclusively on the plump chicks of nesting mutton birds. This dietary shift has driven a corresponding shift in venom. The venom of the Chapel Island snakes is produced in enormous quantities. The average venom yield is more than double that of mainland snakes. While less toxic drop for drop, the sheer volume injected makes them incredibly dangerous. It's an evolutionary trade-off. Instead of evolving higher potency, selection has favored the ability to deliver a massive, overwhelming dose to quickly dispatch their larger prey. So how does this incredible diversity happen at a molecular level? The study of this genetic arsenal is known as venomics. The process begins with what is known as gene recruitment and duplication. It all starts with a gene coding for a normal body protein, like a digestive enzyme. Through a random mutation, a copy of this gene is created. The original gene continues its vital function, but this spare copy is free to accumulate new mutations without harming the snake. Eventually, it is recruited into the venom gland, meaning it begins to be expressed there. Once this duplicated gene is expressed in the venom, it is subject to intense positive selection. Any mutation that makes the resulting protein even slightly more toxic provides a survival advantage. Over millennia, this process transforms a mundane body protein into a deadly toxin. This geographical variation in venom is not just an evolutionary marvel, it is a matter of life and death for the estimated 2.7 million people envenomated each year, leading to over 130,000 deaths and 400,000 cases of permanent disability. The primary treatment is antivenom, and its effectiveness is directly threatened by venom geography. Antivenom production is a century-old technology. It involves milking venom from snakes and injecting small doses into a large host animal, usually a horse. The animal's immune system produces antibodies against the toxins. These antibodies are then purified and formulated into antivenom. Herein lies the critical problem. The antibodies produced are highly specific to the venom used in the immunization process. An antivenom created using venom from Russell's vipers in southern India may be highly effective there, but it can be alarmingly ineffective against a bite from the same species in northern India, where the venom composition is different. This forces doctors into a desperate situation, sometimes leading to treatment failure and death in cases that should have been survivable. Faced with this crisis, scientists are racing to develop a new generation of therapies. The most promising frontier is the development of recombinant antivenoms. Instead of using horses, this approach uses modern biotechnology to produce pure, specific monoclonal antibodies in a laboratory setting. The advantages are enormous. They are safer, more consistent, and more potent. The ultimate dream is a single, universal antivenom that would be effective against the bite of any medically significant snake in the world. For a long time, this was considered science fiction, but recent breakthroughs have brought it into the realm of possibility. Scientists have studied the blood of an amateur herpetologist who has spent years self-immunizing by injecting himself with a wide array of venoms. They discovered several antibodies in his blood that can neutralize toxins from a vast range of snakes from different continents. By analyzing these antibodies, scientists have identified conserved, unchanging regions on the toxins. By targeting these critical sites, a single antibody can neutralize toxins from many different snakes. Researchers believe a cocktail of just 4 or 5 of these broadly neutralizing antibodies could potentially offer protection against the venom of almost any deadly snake. In conclusion, venom geography reveals that a snake's venom is a liquid diary, recording millennia of evolutionary pressures. This intricate variability is both a testament to the power of natural selection and a formidable challenge to modern medicine. Yet, in this challenge lies immense opportunity. By mapping these venomous landscapes, we are paving the way for a revolution in snakebite treatment. The future of antivenom lies not in fields with horses, but in the gleaming bioreactors of the laboratory. The quest for a universal antivenom, once a distant dream, is now an active and achievable goal, reminding us that the deepest biological secrets often have the most profound human consequences. Thanks for tuning in to Fun Facts Online.